So, students, we came to know that light reaction is meant for the synthesis of assimilatory power, that is ATP and NADPH, and that occur in presence of sunlight by the involvement of photosynthetic pigments, and photosynthetic pigments are organized in the form of photosystems located in thylakoid membrane. And in addition to photosystems and various electron carrier complexes located in thylakoid membrane, and also coupling factor, that is CF0, CF1, and they are involved in light phase reactions and involved in the synthesis of assimilatory power, that is ATP and NADPH. Okay, now let us elaborate and what are photosystems and how they are involved in light phase reaction and how they cause that synthesis of assimilatory power here. And we need an elaboration of photosystems, nothing but bundle of photosynthetic pigments, and who discovered photosystems and all. Now let us see photosystems, as you heard about PS1 and PS2, and they are nothing but bundle of photosynthetic pigments with reaction center and light harvesting complex, and they were discovered by Robert Emerson as he experimented in a unicellular green alga, that is chlorella. And now let us follow the details of that experiment conducted by Emerson, and he experimented on chlorella, I said, and chlorella is exposed with red light, and mean the light selected by Emerson is red light, we can say monochromatic light, and he exposed chlorella cells to continuous red light in the first instance, mean red light of only longer wavelength is given to chlorella. Then he observed a sudden drop in photosynthetic activity and named it as red drop. Red drop means a sudden drop in photosynthetic activity in presence of continuous red light in chlorella observed by Emerson, that is red drop. Later, in the second instance, he exposed chlorella cells to red light of both longer and shorter wavelength, that is by giving intermittent light or discontinuous light. So, this time chlorella is provided with both longer and shorter wavelength, and here he observed a sudden enhancement or sudden increase in photosynthetic activity and named it as enhancement effect. Now, as we conclude these two instances, and what is the conclusion given by Emerson as we verified? And as chlorella exposed with continuous red light, that leads to sudden drop in photosynthetic activity because there are two photosystems, but among the two, one photosystem is function only in that light of longer wavelength, whereas the other photosystem is not functioning in presence of longer wavelength. So, among the two photosystems, only one photosystem is in function because of only longer wavelength provided, whereas the other photosystem is non-functioning. That leads to decrease in photosynthetic activity, and that is red drop. But whereas, and in the other instance, chlorella exposed to intermittent light provided with both longer and shorter wavelength, among the two photosystems, one photosystem is function in longer wavelength and the other photosystem is functional in shorter wavelength. A simultaneous supply of both longer and shorter wavelength, so that both the photosystems, they show synergistic action, and that cause sudden enhancement or increase in photosynthetic activity. So, enhancement effect is observed by Emerson. So, based on this observation, Emerson proposed there are two photosystems in higher plants, and that is PS1 and PS2, and these two photosystems, they vary in their reaction center pigment, and the reaction center pigment of PS1, and that is P700, which observe longer wavelength, whereas the reaction center pigment of PS2 and which is P680, and it can able to absorb only shorter wavelength. So, as only longer wavelength provided at that time, only PS1 is in function, that cause drop in photosynthetic activity. Whereas at the time when chlorella are exposed to both longer and shorter wavelength, both the photosystems are in function. So that simultaneous and synergistic functioning of both the photosystems cause enhancement or increase in photosynthetic activity. So based on this observation, he revealed there are two photosystems in higher plants, that is PS1 and PS2. Okay? And now coming to photosystems. What are the components and how they are involved in this photochemical reaction and in light phase mechanism. Okay. Now let us continue the structure of photosystems and the components and how they are involved in light phase reaction. And remaining, we discuss about the structure and components of photosystems, how they are involved in photosynthetic activity, and we will discuss in the next session. Students, as we continue the discussion on photosystems, now let us see and what are photosystems and what are the types of photosystems, what is their structure and function. Now, there are two types of photosystems as concluded by Robert Emerson. They are photosystem 1 and photosystem 2. Based on their discovery only they are named. The first discovered one is photosystem 1 and the later observed or later discovered is photosystem 2. And any photosystem, whether it is photosystem 1 or photosystem 2, any photosystem with two components, and that is reaction center and light harvesting complex. The other name for light harvesting complex is antennae chlorophyll complex. So, the two components of a photosystem are reaction center and light harvesting complex. Reaction center is always with a single chlorophyll A molecule. In the place of reaction center we can see here a single chlorophyll A molecule. If it is the reaction center of photosystem 1 and that is a single chlorophyll A molecule, that is P700, whose absorption maxima is at 700 nanometer. Okay? And whereas the reaction center of PS2 as we see here, that is also the single chlorophyll A molecule, but and it is referred as pigment 680 because its absorption maxima is 680. So, here the reaction center pigment of photosystem 1, that is chlorophyll A of P700 type, and that is function in longer wavelength and absorb longer wavelength of light and get excited and liberate electrons. Such electrons involved in electron transport mechanism and cause the synthesis of NADPH and ATP. And in the same way, the reaction center pigment of photosystem 2, that is single chlorophyll A molecule of P680, whose absorption maxima 680, mean absorb shorter wavelength and get excited and liberate electrons, and such electrons are involved in electron transport, particularly non-cyclic electron transport, and involved in the synthesis of assimilatory power. And now coming to light harvesting complex. Light harvesting complex of any photosystem, it is made of accessory pigments that includes some chlorophyll A molecules also, of course chlorophyll B, carotenes and xanthophylls. So, light harvesting complex contain mainly chlorophyll B, carotenes and xanthophylls in higher plants. Even we can found chlorophyll A molecules also. So, a single chlorophyll A molecule and found in reaction center directly involved in photochemical reaction. Other chlorophyll A molecules as well as chlorophyll B and carotenes and xanthophylls are located in light harvesting complex, the surrounding area of reaction center, and they also trap the solar energy, but they are not involved in photochemical reaction. They shift that solar energy to the reaction center. So, they can be referred as antennae chlorophyll molecules. Okay? And here LHC1 and LHC2, they contain the necessary pigments which act like antennae molecules. So, this particular space is called light harvesting complex or antennae chlorophyll complex, where the necessary pigments are involved. And they not only shift the solar energy, they also cover the reaction center and protect the reaction center from photooxidation or photobleaching. Okay? That is about the photosystems and how they are involved in light phase. As we followed, a single chlorophyll A molecule present in the reaction center area directly involved in photochemical reaction in presence of sunlight get excited and liberate electrons. Such electrons are involved in electron transport and help in the synthesis of NADPH and ATP. That's about photosystems. Now, let us continue the mechanism of light reaction, and that is by the involvement of electron carrier complexes and how it causes photophosphorylation, that is synthesis of ATP by the involvement of coupling factor, that is the detailed mechanism of light phase reactions. And the main important event of light reaction is electron transport. And there are two types of electron transport mechanisms. They are non-cyclic electron transport and cyclic electron transport. Non-cyclic as well as cyclic electron transport, which is the main event of light phase reactions in association with photolysis of water as well as phosphorylation. Students, let us see that non-cyclic electron transport that occur during light phase reactions. And in non-cyclic electron transport, there is the involvement of PS2 and PS1, both the photosystems involved, as well as water molecule that undergo photolysis, splitting in presence of sunlight, and electron carrier complexes like cytochrome B6 and F. In addition, two types of mobile electron carriers also involved, that is plastoquinone and plastocyanin. So, non-cyclic electron transport that occur in higher plants, that is from cyanobacteria up to angiosperms, and that cause evolution of oxygen by splitting of water molecule and is a part of oxygenic photosynthetic light phase, and that include the activity of PS1 and PS2, both the photosystems involved, and involvement of water molecule that undergo splitting, and electron carrier complexes cytochrome B6 and F, as well as the mobile electron carriers plastoquinone and plastocyanin. Okay. Now, and what are the different steps that are involved in non-cyclic electron transport? Let us see. The first step in non-cyclic electron transport is, that is excitation of chlorophyll A pigment, that is reaction center pigment, particularly the chlorophyll A of PS2 reaction center, that is P680 molecule, and it liberate electrons, and that electrons, and they are transported through various electron carrier complexes. But meanwhile, that excited P680 and should attain ground state. For that, it require electrons and it cause splitting of water molecule. So, the first step is excitation of chlorophyll A pigment of PS2, that is a reaction center pigment of PS2 P680. And the next step is, and to compensate that electron loss for P680, photolysis of water, that is splitting of water molecule takes place, and the electrons from that P680 carried through various electron carriers and finally reaches to stroma, and there NADP plus available always is now reduced to NADPH as it received that electrons. Now, and due to that electron transport mechanism, as plastoquinone is also involved in electron transport that cause proton translocation from stroma to lumen, and protons also from water molecules added to lumen. So, proton translocation from stroma to lumen as well as from water to lumen, and that cause proton concentration gradients and later on leads to the synthesis of ATP. So, these are the various steps that occur during non-cyclic electron transport, finally cause the synthesis of assimilatory power, that is NADPH and ATP. Okay. Now, let us follow a detailed mechanism of non-cyclic electron transport. Students, now let us go through the detailed mechanism of non-cyclic electron transport. What are the events and what are the subsequent results? How that non-cyclic electron transport involved in the synthesis of NADPH and ATP. Now, as we follow the steps involved in non-cyclic electron transport, the first step is excitation of chlorophyll A molecule of PS2, that is the reaction center pigment P680. In presence of sunlight, P680 molecule, the chlorophyll A of P680, and absorb sunlight and get excited and liberate a pair of electrons. Okay. And the number of photons utilized for this excitation is 2 photons. Okay, you know photons is the light quanta, light energy utilized, required for the excitation of this chlorophyll pigment. So 2 photons are utilized and 2 electrons are liberated, and that liberated electrons, 2 electrons, they are received by a colorless chlorophyll A molecule present in LHC2, that is pheophytin. So pheophytin is the primary acceptor of electrons from P680 molecule, and from pheophytin the electrons are received by a mobile electron carrier that is located in thylakoid membrane, that is plastoquinone. And plastoquinone, here it take electrons from pheophytin, but it is not only the electron carrier, it also pulls the protons, because plastoquinone reduced to plastoquinol here, and for that reduction process not only electrons from pheophytin, it requires protons also, and the protons are taken from stroma. They are pulled by stroma, the mean pulled from stroma by plastoquinone. So it is an active process we consider. So the active uptake of 2 protons by plastoquinone reduce plastoquinone into plastoquinol. So here all of you once again let us follow. Pheophytin had 2 electrons, and which is the primary acceptor of electrons from excited P680. Now that 2 electrons are received by plastoquinone, and plastoquinone not only take that electrons, it also take protons, and the proton source is stroma. Stroma is always with large number of protons, as we know. So, and here protons, 2 protons are received by or pulled by plastoquinone by active process, okay, and now it reduced to plastoquinol PQH2, right, and immediately undergo oxidation again and liberate that electrons and protons, and that electrons are received by cytochrome B6, and which is a electron carrier in cytochrome complex. But of course here cytochrome complex is only electron carrier, but it do not hold the protons. So this cytochrome complex leave the protons into lumen. So this is very important event we have to follow, students, to understand that Peter Mischel's chemiasmatic hypothesis. So I will repeat once again. Here then the excited P680 liberated 2 electrons, and that 2 electrons received by a primary acceptor, that is pheophytin, located in LHC2, and from the pheophytin the electrons received by plastoquinone, and plastoquinone receive electrons from pheophytin and pulls the protons from stroma by active process and reduce it to plastoquinol, okay, and again immediately get oxidized. And here due to that oxidation of plastoquinol, again that electrons and protons here, and they entered into cytochrome complex, but cytochrome complex receive only electrons but not protons. So the protons left by cytochrome complex into the lumen part. So here proton translocation occur as plastoquinone is both electron and proton carrier, as cytochrome complex is only the electron carrier but not the proton carrier. So such protons which are taken by plastoquinone from stroma and they are left to the lumen are translocated to lumen, as cytochrome complex is only electron carrier but do not take the protons. So that is the reason why proton translocation from stroma to lumen occur during non-cyclic electron transport. And meanwhile we have to follow one more event here, and here P680 molecule is in excited condition, we said, already liberated 2 electrons, but within nanosecond it should attain ground state, and to attain ground state it need electron donor. So that excited P680, which is a strong oxidant now, and it stimulate that splitting of water molecule to get electrons through OEC complex, and the full form of OEC, oxygen evolving complex, and it stimulate and motivate that splitting of water molecule as it is stimulated by excited P680. So water split into 2 electrons, 2 protons, and liberate half molecule of oxygen, and that 2 electrons through OEC received by excited P680 and attain ground state. So, the electron loss of P680 is compensated by electrons liberated by the splitting of water molecule, and this particular event, splitting of water, is called photolysis of water, mean the lysis, that is breakdown of water in presence of sunlight. Of course, and just recollect, as we take water in the bowl and expose it to sunlight, is that water molecule split in the outside environment? No. So, here and in presence of sunlight, water splits, that is due to excited chlorophyll pigment. So, here the relation is, in presence of sunlight, the chlorophyll P680 get excited, that excited P680 is the motivator and stimulate the splitting of water molecule into electrons, protons and oxygen through OEC complex. Now, the electrons through OEC received by P680 and the electron loss is compensated. Okay, by whom? The electrons of water molecule. Now, and what happened to that electrons received by cytochrome complex as we continue? The electrons from cytochrome complex, they are received by plastocyanin, a copper containing mobile electron carrier nearer to PS1, and already the reaction center pigment, that is chlorophyll A of P700 type, and which is the reaction center pigment of PS1, already get excited and ready to receive the electrons. So, the electrons from plastocyanin received by the chlorophyll A P700 in the reaction center of PS1, and already the liberated electrons of chlorophyll A of P700 type now received by an unknown acceptor in LHC1, and that unknown acceptor give the electrons to iron containing electron carrier, iron sulfur protein, that is ferridoxin, and from the ferridoxin the electrons are received by a catalyst here, that is the enzyme ferridoxin NADP reductase enzyme, or simply NADP reductase enzyme, which is located of course in the outer surface of thylakoid membrane towards the stroma part, and that receive the electrons, and meanwhile protons are supplied by stroma, as already we said, and stroma always with protons. So, in presence of the electrons and the protons from stroma, NADP plus in the stroma reduced to NADPH, and that is synthesis of reducing power due to electron transport, that is non-cyclic electron transport mechanism. Okay. Now, as we verify this non-cyclic electron transport flow, here the electrons of P680 received by P700 and the electrons of P700 received by NADP plus, that we observe, but the electron loss of P680 is compensated by water molecule. So, here as we follow tally accounting, okay, as we follow tally accounting, here the electrons of water molecule received by PS2, that is P680 molecule, and from PS2, that is pheophytin, and pheophytin gives the plastoquinone, and plastoquinone cytochrome B6, and then plastocyanin, and plastocyanin give rise to gives that electrons to P700. From P700 the electrons, they are through unknown and ferridoxin, and finally received by NADP plus. So, as we follow the electron flow, the electron loss of PS2 compensated by water, the electron loss of PS1 compensated by PS2, and finally electrons received by NADP plus. Finally, the loser of electrons is water molecule. So, as we follow this kind of electron transport, this is non-cyclic electron transport. No electrons back to their original home we followed. Electrons of water to PS2, PS2 to PS1, PS1 to NADP plus. So, it is a kind of non-cyclic electron transport, and the scientists called Hill and Bendall, and they referred this kind of electron transport as jet scheme of electron transport. As we follow, and this particular electron flow that we follow, that resemble the alphabet letter Z as explained by Hill and Bendall. So, this is also called jet scheme of electron transport, and we will discuss in the next session.